This is the video for experiment 5, assaying for specific metabolism activities of microorganisms. Firstly, since we know that microorganisms have several central metabolic pathways involve the flow of energy and carbon through the cell, since different microbes produce various biochemical product by using the nutrients from the medium. Therefore, there are several methods used to determine the type of microbes based on the substance consumed by the microbe. 4 parts involved in this experiment, which is acid and gas productions from carbohydrate fermentations, and Vogel's post-Calvert test, methylene red test, and the last one is citrate utilization test. For the first test, acid and gas productions from carbohydrate fermentations, it is the microbes convert carbohydrate into organic acid and either with the formations of gas bubble or without. For the next one is Vogel's press-covered test. It is a test to determine the formations of acetate methylate carbinol from glucose fermentations by an organism. And then the methylate rate test is the test to determine the formations of stable acids such as lactic acid from converting a glucose by a microbes as an end product. And then the last part is citrate utilizations, which is a test to differentiate among the gram-negative bacillus in the family Enterobacteriaceae based on the formations of color change from green to royal blue. Therefore, the objective for this experiment is to examine several specific metabolic activities of microorganisms that are frequently used to identify bacteria species, and the second one is to detect metabolic pathway that differentiate microorganisms from this experiment.
The procedure began by adding 1 ml of specific carbohydrates to purple broth or fennel red broth tube, which already contained a protein source for bacterial growth and a pH indicator. The purple broth base had from crescel purple turning from purple to yellow at acidic pH, while fennel red changed from red to yellow under acidic conditions. The tubes were labeled accordingly and the test bacteria were inoculated into carbohydrate supplemented tubes, and the tubes were then incubated at 37 degrees Celsius for 24 to 48 hours. After incubation, the tube colors were observed with yellow indicating acid production. The room tubes were checked for gas production evident through partial or complete filling. Results were recorded based on the observed color changes and gas production. The procedure began by inoculating tubes of MRVP broth with each test culture and labeling the tubes. The inoculated tubes were then incubated at 37 degrees Celsius for 24 to 48 hours. After incubation, 1 ml of culture was aseptically pipetted into a clean test tube. To this, 0.5 ml of Barrett's reagent A and 0.5 ml of Barrett's reagent B were added. The mixture was shaken vigorously for 30 seconds and the tube were observed for the gradual formation of a pink to red color indicating a positive test for acetone. Some cultures show the reaction within 1 minute while others took 1 to 2 hours. The results were recorded accordingly. The leftover broth from the cultures used in the Vogel's post-cow test was utilized for the methyl red test. If the Vogel's post-cow test had not been conducted, tubes of MRVP medium were inoculated with each culture and incubated at 37 degrees Celsius for 24 to 48 hours. Methyl red solution was added to the cultures and immediate red reactions indicate mixed acid fermentation, while yellow reaction indicate a negative test. Results were recorded accordingly. Firstly, agar slants of Siemens citrate medium were inoculated with each test microorganism. The tubes were then incubated at 37 degrees Celsius for 24 to 48 hours. After the incubation period, the color of the tubes was observed. A royal blue color indicates a positive test for citrate utilization, while a green color indicates a negative test. The results were recorded accordingly.
This section is the result shown for acid and gas productions from carbohydrate fermentations. As we seen from the diagram, the sample B is located on the right-hand side, which able to determine since there is a formation of gas bubble within the dimer tube. Whereas for the sample A, which is located on the left-hand side, does not have the formations of gas bubble within the dimer. Therefore, this represents that the sample B is ascarical chloride due to the formations of acidic end product and gas bubble from the specific carbohydrate substance. Whereas for sample A, which is shown a negative reaction, is determined as serratia maceracein due to the absence of gas bubble within the term. For this section is the result for Woger's press cover test. Based on the diagram, there is an observation made that there is two color differences from test tube on the right hand side and the test tube from the left hand side. These the result sections for Woger's press covered test. Based on the result obtained from the experiment, the right hand side test tube show a positive result which the color changed from pink to red color. This show the formation of acetone and the microbes is determined as Enterobacter aerogenes due to the microorganism involved in the butadiene pathway and the formations of butadiene as a. Beside that, for the left-hand side test tube show a negative result, which means the formations of acetone is absent and the involved microbes is known as ascarical chloride, which does not undergo the butadiene pathway in the metabolism activity. Furthermore, the methyl carbonyl formed from the butanediol pathway able convert into diacetic with the presence of alpha-naphthone strong alkali such as KOH and the oxygen from atmospheric surface. Therefore, the Walker's press covered test able to determine based on the color changes, which is from pink to red color appear at the end of. Citrate utilization is a process where the change of color from green to royal blue due to the change of pH. So from the diagram is able to observe that there is no observations provided. So from the table is able to observe there is no reactions, which is it indicate negative reactions, doesn't have color change. So for the methylene red test is a process of color change occur from yellow to red due to deformations of acidic product from the microorganism close to pH value less than. So the result is tabulated on the table. For the microorganism Cassella phenomena, the reaction is remain of yellow color, whereas the ascarical chloride turns from yellow to red, which indicate there is a formation of acidic.
Question number 1. Your microorganism grows in the tube with glucose, showing an increase in turbidity, but the tube does not turn yellow. Why? The absence of yellow color change despite increased turbidity suggests that the microorganism may produce metabolic byproducts like organic acids without generating the acidic end products that typically turn the indicator yellow. Question number 2. What gas other than carbon dioxide might be produced and collected in the drum tube? Besides carbon dioxide, other gases such as hydrogen might be produced and collected in the drum tube during fermentation. Question number 3. How could you detect acid and gas production using a solid medium? Acid and gas production can be detected using a solid medium by incorporating a pH indicator, example phenol red, into the medium. Acid production results in a color change and gas production is observed as bubbles or fissures in the agar. Question number 1. If the Voges-Proskauer test is run too early or too late, it will be negative even the microorganism is carrying out the butanediol fermentation pathway. Why? Running the Voges-Proskauer test too early or too late may yield a negative result because the intermediate products required for a positive reaction may not be present in the correct concentration. Question number 2. Why is the Voges-Proskauer test important in microbiome taxonomy? The Voges-Proskauer test is crucial in microbiota as it helps differentiate bacteria undergoing mixed acid fermentation positive result from those utilizing the butanediol fermentation pathway negative result, aiding in microorganism identification and classification. Question number 1. Can a microorganism carry out both the butanediol and mixed acid fermentations? Yes, a microorganism can carry out both the butanediol and mixed acid fermentations, resulting in varying test outcomes. Question number 2. Can a microorganism be both methyl red and Voges-Proskauer positive? Explain. No, a microorganism cannot be both methyl red and Voges-Proskauer positive. The tests represent different fermentation pathway where methyl red detects mixed acid fermentation while Voges-Proskauer detects butanediol fermentation. Question number 1. Why does the pH of Siemens citrate agar increase if citrate is utilized? The pH of Siemens citrate agar increases when citrate is utilized because the breakdown of citrate results in the production of alkaline byproducts, leading to a rise in pH. Question number 2. The microorganism grows on Siemens citrate agar where the slanted portion is blue but the bud remains green. Explain. If the microorganism grows on Siemens citrate agar and the slanted portion is blue while the bud remains green, it suggests that citrate utilization occurs only in the slanted upper portion and the alkaline byproduct did not diffuse to the lower bud region. Question number 3. How would the MVAT test differentiate Escherichia coli from Enterobacter aerogenes? The MVAT test differentiates Escherichia coli from Enterobacter aerogenes based on various results. E. coli typically show positive result for indole production and Voges-Proskauer, negative for methyl red and citrate utilization, while Enterobacter aerogenes may display the opposite pattern.